Excretory product and their elimination. In this chapter, we will read about the human excretory system, urine formation, function of tubules, mechanism of concentration of the filtrate, regulation of the kidney function, mixturation, role of the other organs in excretion, disorder of the excretory systems. Okay. Animals accumulate ammonia, urea, uric acid, carbon dioxide, water, and the ions like sodium, potassium, chlorine, phosphate, sulfate, etc., either by the metabolic activities or by the other means like excess ingestion. These substances have to be removed totally or partially. In this chapter, you will learn the mechanism of elimination of these substances with the special emphasis on the common nitrogenous waste. Ammonia, urea, and the uric acid are the major form of nitrogenous waste excreted by the animals. Ammonia is the most toxic form and requires large amount of water for its elimination, whereas uric acid, being the least toxic, can be removed with minimum loss of water. The process of excreting ammonia is called ammonotalism. Many bony fishes, aquatic amphibians, and the other insects are ammonotelic in nature. Ammonia, as it is readily soluble, is generally excreted by the diffusion across body surfaces or through gill surfaces in fishes, as in ammonium ions. Kidneys do not play any significant role in its removal. Terrestrial adaptation necessitated the production of lesser toxic nitrogenous wastes like urea and the uric acid for the conservation of water. Mammals, many terrestrial amphibians, and the marine fishes mainly excrete urea and are called ureotelic animals. Ammonia produced by the metabolism is converted into urea in the liver of these animals and released into the blood, which is filtered and excreted out of the kidneys. Some amount of urea may be retained in the kidney matrix of some of these animals to maintain a desired osmolarity. Reptiles, birds, land snails, and the insects excrete nitrogenous waste as a uric acid in the form of pellet or the paste with a minimum loss of water and are called ureotelic animals. A survey of animal kingdom presents a variety of excretory structures. In the most of the invertebrates, these structures are simple tubular forms, whereas vertebrates have complex tubular organs called the kidney. Some of these structures are mentioned here. Protonephridia or flame cells are the excretory structures in Platyhelminthes, that is flatworms, example Planaria, rotifers, some annelids, and the cephalochordate, that is Amphioxus. Protonephridia are primarily concerned with the ionic and the fluid volume regulation, that is osmoregulation. Nephridia are the tubular excretory structure of earthworms and the other annelids. Nephridia help to remove nitrogenous waste and maintain a fluid and ionic balance. Malpighian tubules are the excretory structures of the most of the insects, including cockroaches. Malpighian tubules help in the removal of nitrogenous waste and osmoregulation. Antennal glands or the green glands perform the excretory function in the crustaceans like prawns. Human excretory system. In humans, the excretory system consists of a pair of kidneys, one pair of ureters, and a urinary bladder and urethra. Kidneys are the reddish brown, bean-shaped structure situated between the levels of the last thoracic and the third lumbar vertebra, close to the dorsal inner wall of the abdominal cavity. Each kidney of an adult human measure 10 to 12 cm in the length, 5 to 7 cm in the width, 2 to 3 cm in the thickness, with an average weight of 120 to 170 gram. Towards the center of the inner concave surface of the kidney is a notch called the hilum, through which ureter, blood vessels, and the nerves enter. Inner to the hilum is a broad funnel-shaped space called the renal pelvis, with projections called the calyces. The outer layer of the kidney is a tough capsule. Inside the kidney there are two zones, an outer cortex and inner medulla, and the medulla is divided into a few conical masses, that is medullary pyramids, projecting into the calyces, the singular calyx. The cortex extends in between the medullary pyramids as the renal columns called the columns of Vertini. Each kidney has nearly 1 million complex tubular structure called nephrons, which are the functional units. Each nephron has two parts, the glomerulus and the renal tubule. Glomerulus is a tuft of capillaries formed by the afferent arteriole, a fine branch of renal artery. Blood from the glomerulus is carried away by an efferent arteriole. The renal tubules begin with a double-walled cup-like structure called Bowman's capsule, which encloses the glomerulus. Glomerulus along with the Bowman capsule is called the Malpighian body or the renal corpuscle, and the tubule continues further to form a highly coiled network, that is proximal convoluted tubule. that is PCT. A hairpin shaped Henle's loop is the next part of the tubule which has a descending and an ascending limb. The ascending limb continues as another highly coiled tubular regions called the DCT, that is distal convoluted tubule. The DCTs of many nephron open into an straight tube called the collecting duct, many of which converge and open into the renal pelvis through the medullary pyramids in the calyces. The Malpighian corpuscles, PCT, and the DCT of the nephron are situated in the cortical region of the kidney, whereas the loop of Henle dips into the medulla. In majority of the nephrons, the loop of Henle is too short and extend only very little into the medulla. Such nephrons are called cortical nephrons. In the sum of the nephrons, the loop of Henle is very long and runs deep into the medulla. These nephrons are called juxtamedullary nephrons. The efferent arteriole emerging from the glomerulus forms a fine capillary network around the renal tubule called the peritubular capillaries. A minute vessel of this network runs parallel to the Henle's loop, forming a U-shaped vasa recta. Vasa recta is absent or highly reduced in the cortical nephron. Now, urine formation. Urine formation involves three main processes, namely glomerular filtration, reabsorption, and secretion that takes place in different parts of the nephron. The first step in the urine formation is the filtration of blood, which is carried out by the glomerulus and is called glomerular filtration. On an average, 1100 to 1200 ml of the blood is filtered by the kidney per minute, which constitutes roughly 1/5 of the blood pumped out by each ventricle of the heart in a minute. The glomerular capillary blood pressure causes filtration of blood through three layers, that is the endothelium of glomerular blood vessels and the epithelium of Bowman's capsule and a basement membrane between these two layers. The epithelial cells of the Bowman's capsule called podocytes are arranged in an intricate manner so as to leave some minute spaces called filtration slit or the slit pores. Blood is filtered so finely through these membranes that almost all the constituent of the plasma except proteins pass on to the lumen of the Bowman's capsules. Therefore, it is considered as the process of ultrafiltration. The amount of filtrate formed by the kidneys per minute is called glomerular filtration rate, that is GFR. GFR in a healthy individual is approximately 125 ml per minute, that is 180 liter per day. The kidneys have built-in mechanism for the regulation of glomerular filtration rate. One such efficient mechanism is carried out by the juxtaglomerular apparatus, that is JGA. JGA is a special sensitive region formed by the cellular modification in the distal convoluted tubule and the afferent arteriole at the location of their contact. A fall in GFR can activate the JJ cells to release renin, which can stimulate the glomerular blood flow and thereby the GFR back to the normal. A comparison of the volume of the filtrate formed per day, that is 180 liter per day, with that of the urine release, that is 1.5 liters, suggests that nearly 99% of the filtrate has to be reabsorbed by the renal tubules, and this process is called reabsorption. The tubular epithelia cells in the different segment of the nephron perform this either by the active or the passive mechanism. For example, substances like glucose, amino acid, sodium, etc. in the filtrate are reabsorbed actively, whereas the nitrogenous waste are absorbed by the passive transport. Reabsorption of water occurs passively in the initial segment of the nephron. During the urine formation, the tubular cells secrete substances like H+, K+, and ammonia into the filtrate. Tubular secretion is also an important step in the urine formation as it helps in the maintenance of ionic and the acid-base balance of the fluid. Functions of the tubules. PCT. Proximal convoluted tubule. PCT is lined by the simple cuboidal brush border epithelium, which increases the surface area for the reabsorption. Nearly all the essential nutrients and the 70 to 80% of the electrolytes and the water are reabsorbed by this segment. PCT also helps to maintain the pH and the ionic balance of the body fluid by the selective secretion of hydrogen ions, ammonia, and the potassium ions into the filtrate and by the absorption of bicarbonate from it. Henle's loop. Reabsorption is the minimum in its ascending limb. However, this region plays a significant role in the maintenance of the high osmolarity of the medullary interstitial fluid. The descending limb of the loop Henle is permeable to the water but almost impermeable to electrolytes. This concentrates the filtrate as it moves down. The ascending limb is impermeable to water but allows transport of electrolytes actively or the passively. Therefore, as the concentrated filtrate passes upward, it gets diluted due to the passage of electrolyte to the medullary fluid. DCT, that is distal convoluted tubule. Conditional reabsorption of the sodium and water takes place in this segment. DCT is also capable of reabsorption of bicarbonate and the selective secretion of hydrogen and potassium ions and the NH3 to maintain the pH and the sodium potassium balance in the blood. Collecting duct. This long duct extends from the cortex of the kidney to the inner part of the medulla. Large amount of the water could be reabsorbed from this region to produce a concentrated urine. This segment allows passage of a small amount of urea into the medullary interstitial to keep the osmolarity. It plays an important role in the maintenance of pH and ionic balance of the blood by the selective secretion of H plus and the K plus ions. Mechanism of concentration of the filtrate. Mammals have the ability to produce a concentrated urine. The Henle's loop and the vasa recta play a significant role in this, and the flow of filtrate in the two limbs of Henle loop is in the opposite direction and thus forms a counter current, and the flow of the blood through the two limbs of the vasa recta also in a counter current pattern. The proximity between the Henle loop and the vasa recta as well as the counter current in them help in maintaining and increasing osmolarity towards the inner medullary interstitium, that is from 300 millismoliter in the cortex to about 1200 millismoliter in the inner medulla. This gradient is mainly caused by the NaCl and the urea. NaCl is transported by the ascending limb of the Henle loop, which is extended by the descending limb of the vasa recta. NaCl is returned to the interstitium by the ascending portion of the vasa recta. Similarly, a small amount of urea enters the thin segment of the ascending limb of the Henle's loop, which is transported back to the interstitium by the collecting tubule. The above described transport of the substances facilitated by the special arrangement of Henle loop and the vasa recta is called counter current mechanism, and this mechanism helps to maintain a concentration gradient. In the medullary interstitium, presence of such interstitial gradient helps in an easy passage of water from the collecting tubule, thereby concentrating the filtrate urine, that is urine. Human kidneys can produce urine nearly 4 times concentrated than that of the initial filtrate formed. Regulation of kidney function. The functioning of the kidney is efficiently monitored and regulated by a hormonal feedback mechanism involving the hypothalamus, JGA, and to a certain extent the heart. Osmoreceptors in the body are activated by the changes in the blood volume, body fluid volume, and the ionic concentration. An excessive loss of fluid from the body can activate this receptor, which stimulate the hypothalamus to release ADH, that is antidiuretic hormone, or the vasopressin from the neurohypophysis. ADH facilitates the water reabsorption from the latter part of the tubule, thereby preventing the diuresis. An increase in the body fluid volume can switch off the osmoreceptors and suppresses the ADH release to complete the feedback. ADH can also affect the kidney function by its constrictory effect on the blood vessels. This causes an increase in the blood pressure, and increase in the blood pressure can increase the glomerular blood flow and thereby GFR. The JGA plays a complex regulatory role. A fall in the GFR, that is glomerular blood flow, that is glomerular blood pressure, that is GFR, can activate the JG cells to release the renin, which converts angiotensinogen in the blood to angiotensin 1 and further to angiotensin 2. Angiotensin 2, being a powerful vasoconstrictor, increases the glomerular blood pressure and thereby GFR. Angiotensin 2 also activates the adrenal cortex to release aldosterone. Aldosterone causes reabsorption of sodium and the water from the distal part of the tubule, and this also leads to an increase in the blood pressure and the GFR. This complex mechanism is generally known as the renin-angiotensin mechanism. An increase in the blood flow to the arteria of the heart can cause the release of atrial natriuretic factor, that is ANF. ANF causes vasodilation, dilation of blood vessels, and thereby decreases the blood pressure. ANF mechanism therefore act as a check on the renin-angiotensin mechanism. Micturition. Urine formed by the nephron is ultimately carried to the urinary bladder, where it is stored till a voluntary signal is given by the central nervous system, that is CNS. This signal is initiated by the stretching of the urinary bladder as it gets filled with urine. In response, the stretch receptor on the walls of the bladder sends signals to the CNS. The CNS passes on the motor messages to initiate the contraction of smooth muscles of the bladder, and simultaneous relaxation of urethral sphincter causes the release of urine. The process of release of urine is called micturition, and the neural mechanism causing it is called the micturition reflex. An adult human excretes on an average 1 to 1.5 liter of urine per day. The urine formed is a light yellow colored watery fluid which is slightly acidic, that is pH 6, and has a characteristic odor. On an average, 25 to 30 gram of urea is excreted out per day. Various conditions can affect the characteristics of urine. Analysis of urine helps in the clinical diagnosis of many metabolic disorders as well as malfunctioning of the kidney. For example, presence of glucose, that is glucoseuria, and ketone body, that is ketonuria, in the urine are indicative of diabetes mellitus. Role of other organs in the excretion. Other than kidneys, lungs, liver, and skin also help in the elimination of excretory waste. Our lungs remove large amount of CO2, that is approximately 200 ml per minute, and also a significant quantities of water every day. Liver, the largest gland in our body, secrete bile-containing substances like bilirubin, biliverdin, cholesterol, degraded steroidal hormones, vitamins, and the drugs. Most of these substances ultimately pass out along with the digestive wastes. The sweat and the sebaceous gland in the skin can eliminate certain substances through their secretion. Sweat produced by the sweat glands in the watery fluid containing NaCl, a small amount of urea, lactic acid, etc. Though the primary function of sweat is to facilitate a cooling effect on the body surface, it also helps in the removal of the wastage mentioned above. Sebaceous gland eliminates certain substances like sterols, hydrocarbons, and the waxes through the sebum, and this secretion provides a protective oily covering for the skin. Do you know that a small amount of nitrogen waste could be eliminated through saliva too? Disorder of the excretory system. Malfunctioning of kidney can lead to the accumulation of urea in a blood condition called uremia, which is a highly harmful and may lead to the kidney failure. In such patient, urea can be removed by a process called hemodialysis. During the process of hemodialysis, the blood drained from a convenient artery is pumped into a dialyzing unit called the artificial kidney. Blood drained from the convenient artery is pumped into the dialyzing unit after adding an anticoagulant like heparin. The unit contains a coiled cellophane tube surrounded by a fluid, that is dilation fluid, having the same composition as that of the plasma except the nitrogenous waste. The porous cellophane membrane of the tube allows the passage of molecules based on the concentration gradient. As nitrogenous waste are absent in the dialysing fluid, these substances freely move out, thereby clearing the blood. The cleared blood is pumped back to the fluid to the body through a vein after adding antiheparin to it. This method is a boon for the 1,000 uremic patients all over the world. Kidney transplantation is the ultimate method for the correction of acute renal failures, that is kidney failure. A functioning kidney is used in the transplantation from a donor, preferably a close relative, to minimize its chances for the rejection by the immune system of the host. Modern clinical procedures have been increased the success rate of a complicated technique. Renal calculi, stone or the insoluble mass of crystallized salt, that is oxalate, formed within the kidney. Glomerulonephritis, inflammation of glomeruli of the